Hello guys, I am Shubham from Show Tutorials. This video is about the CSIR NET June 25 Part B. Okay. इस सीरीज के प्रीवियस वीडियो में हमने पार्ट बी के 10 क्वेश्चन सॉल्व किए. Okay. So this is the continued in which the remaining questions we will be solving. Okay. So before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from our side, but it will help us grow. So the questions which are solvable based on the syllabus we have covered on our channel, okay, and ऐसे क्वेश्चन जो हम easily solve कर सकते हैं, that I am showing you. Got it? Using the previous knowledge or logical base, right? So first question, which one of the following statements about glycosaminoglycans is incorrect? Okay, incorrect हमें देखना है. GAGs are distinguished by their sugars. Okay, the type of linkage between the sugars and number and location of sulfate groups. So yes, ये correct है, right? This is correct. Sugars present, linkage type between the sugars and sulfate group का number and location. They are one of the most anionic molecules made by animal cells. ये भी correct है. Okay, anionic means negative charged, right? Negative charged. Why? Because they contain sulfate group and carboxyl group, right? Next point, they are branched polysaccharides. Wrong. Okay, they are linear. So GAGs linear होते हैं, not branched. Usually made up of repeating units of sulfated amino sugar and a uronic sugar. Okay, linear होते हैं, not branched. Severe loss of GAGs can lead to premature aging. ये correct है. So इसका answer, the point which is incorrect, that is option 3. Next question, which one of the following does not promote aging in humans? Got it? Aging promote क्या नहीं करेगा इसमें से? Increased oxidative stress. Okay, see, oxidative stress अगर increase हुआ, aging promote होगा. Okay, so this will promote aging, right? Then defects in DNA repair enzymes. This will also, मतलब हम बोल सकते हैं, like as the age increases, DNA repair enzymes defective work कर सकते हैं, right? Defective functional it can cause. So this will promote aging. Got it? Next, activation of telomerase. Okay, अभी telomerase जो है telomeres का रिप्लेशन करते हैं, right? Telomerase basically they act on telomeres, right? What are telomeres? Chromosome ends, right ना? Chromosome ends को telomeres बोलते हैं. तो DNA replication में हमने देखा है end replication problem, so that is solved by telomerase. Got it? So telomere shortening, that is related to aging, right? मतलब जैसे जैसे aging होता है वैसे telomere short होता है, chromosome ends get short, but if the telomerase is activated, so that will be reverse of aging, so this will not promote aging. Okay, not promote aging. तो ये आंसर होगा. Got it? Next point, reduce biosynthesis of leukotrienes. अभी ल्यूकोट्राइन्स का काम क्या है? इन्फ्लेमेशन. inflammation. Okay, leukotrienes and prostaglandins inflammation करते हैं. So we can relate this option to tissue injury, right? Tissue injury से हम relate कर सकते हैं. How that leukotrienes अगर increased है तो inflammation ज़्यादा होगा. I will add I here for inflammation will be more. Means it will do the tissue injury, right? Tissue injury will be more. So if leukotrienes are reduced, okay, as this option is saying, so inflammation कम होगा. Okay, so this will promote aging, right? This will do the aging, and this will not do the aging. Anti-aging. Okay, so ये भी आंसर हो सकता है, right? This can be the answer, but most appropriate statement is 3. Why? Because telomerase activation, this is used for reversal of aging. Anti-aging treatment है ना, उसमें भी इसका यूज होता है टेलोमरेज एक्टिवेशन, एंड बहुत रिसर्च चल रहा है इसमें भी, राइट? लॉट ऑफ रिसर्च गोइंग ऑन इन दिस एरिया. ऑल्सो द कैंसर सेल्स, if you know, cancer cells have high amount of telomeres. Okay, that is why they have the ability to divide continuously, right? So aging उसमें नहीं है, that is reversal of aging. So most correct statement, accurate statement is this activation of telomeres. Got it? We can also say that the option 4 है ना, reduced biosynthesis of leukotrienes, this is more related to immunity, right? So ये vary करेगा depending on person to person. This will vary, but this is fixed. ये जो point है activation of telomerase, this is fixed. मतलब इससे aging reversal होगा ही, right? जो cell की activity है enhance होगी. Got it? As this is already proved by cancer cells. Next question. A student exposed a plant to a herbicide that blocks electron transport both in photosystem 2 and photosystem 1. What is the likely outcome? Okay, electron transport को block किया है. Now this is about photosynthesis. Okay, we have not seen on our channel, but PYQ series में हमने discuss किया है, because this is important, right? Very direct question. When electron transport is blocked, neither ATP nor NADPH would be produced. Okay, इलेक्ट्रॉन ट्रांसपोर्ट ही अगर नहीं हो रहा है तो ATP भी नहीं बनेगा और NADPH भी प्रोड्यूस नहीं होगा. ओके, डायरेक्ट क्वेश्चन है, वेरी इजी. Which one of the following molecule is transported into enterocytes from intestinal lumen by GLUT5? Okay, very repeated question, right? GLUT5, just remember fructose, right? Fructose को transport करता है into enterocytes. Enterocyte means stomach cells from lumen. So here I have attached this picture of glutes, right? Glucose transporter. Okay, make a screenshot of this, very important. So here you can see GLUT5, intestinal epithelium, dietary fructose uptake from the apical side. Okay, and जो insulin response show करता है, that is GLUT4, right, which causes uptake of glucose when glucose is high in blood. GLUT3 is present on neuronal cells. GLUT2 liver cells contains a double amount of glucose than other cells, and GLUT1 erythrocytes. So basically these GLUTs do the facilitated diffusion of glucose. Okay, पैसिव ट्रांसपोर्ट हमने सेल बायोलॉजी में देखा है. नेक्स्ट क्वेश्चन, अ ट्रांसक्रिप्ट ऑफ अ जीन इज शोन बिलो. ओके, जीन दिखाया है विथ लाइंस शोइंग इंट्रॉन and rectangles showing exons. So these are the exons. These are the exons. Exon 1 and exon 2. A mutation from T at the 6th nucleotide position to A in the first exon of the transcript is isolated. Okay, so mutation हुआ है. You can see T is changed to A. Got it? Which one of the following options accurately describes the outcome of this mutation? Now see carefully, जो wild type है and mutant है ATG. ओके, अभी अगर हमने RNA देखा AUG, this is basically start codon, right ना, start codon. And जो next है TAT and that is changed to TAA. So TAE is what? Replace T by U again, UAA. So this is a stop codon, right ना, ये stop codon है. And that means gain of function no, loss of function yes, no change no, formation of an altered protein. Altered protein बनने का चांस ही नहीं है यहाँ पे, because this is start codon and this is changed to stop codon. So no, protein cannot be formed. How the protein will be synthesized? Because start codon के बाद direct stop codon है. So this is the loss of function. Option number 2. Next, 2 temperature sensitive mutants of Escherichia coli were mated with each other and none of the resulting X conjugates showed the wild type phenotype. Okay, very important, wild type phenotype देखने को नहीं मिला. Okay, does not show the wild type. These mutations are, so basically very easy. These questions are also highly repeated. Complementation test. Okay, complementation test से क्वेश्चन है. What is complementation? One gene complements other, right? Okay, I am telling you how you can remember. One gene complements other means अगर same gene mutated है, then there will be no wild type phenotype. Okay, same gene में अगर mutation है तो wild type phenotype नहीं दिखेगा. And when different genes mutated, we will see that they will complement each other and wild type phenotype will be seen. Okay, so when I say same gene, that is alleles of same gene. इस वजह से wild type phenotype देखने को नहीं मिला. So option 1 का आंसर. Got it? Drosophila melanogaster has 4 pairs of chromosomes. Okay, there is no recombination in males of the species, right? Recombination nahi hai. What is the number of possible combinations of chromosomes in gametes of males? Number of possible combinations पूछा है, तो basically n is equal to 4 है. इसका combination we can find 2n. Got it? 2 raised to 4. That gives 16. Option 3. Easy. This is also very easy question. If you know, if you have the basic idea also, then you can solve. According to the Oparin-Haldane hypothesis, which one of the following statements best describes the Earth's early atmosphere? Okay, rich in oxygen and nitrogen? No. Oxygen पहले present नहीं था. Composed primarily of carbon dioxide? Oxygen? No. Oxygen was not present. Reducing atmosphere? Yes, with gases such as methane and ammonia. Correct है. Similar to the current atmosphere of the Earth? No. Option number 3 correct है. Right, gases present थे, methane and ammonia. Reducing environment था. The Krylux system is used to successfully delete a gene specifically in T cells in mice. Okay, Krylux system है. Which one of the following option is correct? Gene को delete करना है, right? Let's see the option. LOXP recombination sequences were introduced in tandem at the 5' end of the gene. I will tell you. This is a target gene. Okay. This is a target gene. अभी इसको हमें delete करना है. So this is a 5' side and this is a 3' side. यहाँ पे हमें LOXP sites को insert करना होगा. 5' and 3', both the sides, where recombinase will come and act. Okay, recombinase we can show as CSIR. Okay, so this is Cree. Cree recombinase will come and cut there. Got it? That much you have to remember for this question. So in this first option it is written 5' end पर ही हमें introduce करना है. So that will be wrong. Second, LOXP recombination sequences were introduced at the 3' end. सिर्फ 3' पर end करके चलेगा? No. Third option, mutant LOXP sites were introduced on either side of the gene. Okay, gene के बाहर either side, and mice were infected with bacteriophage P1 to provide Cree recombinase. Okay, gene को flank करना होगा. Either side of the gene means what? Only one side of the gene. So this is also wrong. Next option, mice containing LOXP sites on both the 5' and 3' ends of the gene were mated with the mice that are transgenic. Okay, transgenic है for Cree recombinase under control of an inducible promoter. Very correct. Okay, very correct. See, मतलब Cree recombinase को भी वहाँ पे प्रमोटर के कंट्रोल के अंडर इंसर्ट किया है, राइट? सो दैट इज कंट्रोल्ड बाय अ इंड्यूसिबल प्रमोटर. Got it? So 4 ऑप्शन करेक्ट है. नेक्स्ट क्वेश्चन, the levels of which one of the following parameters of blood are measured to check cardiac damage? Very easy. Cardiac damage basically heart muscle hai, and the enzyme that is used is creatine kinase, okay, or creatine phosphokinase, CPK और CK. बेसिकली ये मसल डैमेज शो करता है, ओके, एंड हार्ट इज ऑल्सो अ मसल. गॉट इट? वेरी डायरेक्ट. लास्ट वन, which one of the following would be most suitable to study early embryogenesis event using live microscopy of unstained C. elegans embryos? ये भी you have to remember that unstained C. elegans embryos का live microscopy देखना है, and that too of early embryogenesis event. तो ऑप्शन 3 जो है differential interference contrast microscope is used. Got it? DIC microscopy. So confocal microscopy, fluorescence microscopy, and this uses stain. Okay, stain. तो अनस्टेन के लिए हम यूज नहीं कर सकते. एंड सुपर रिजोल्यूशन जो है, दिस इज नॉट फॉर लाइव, राइट? लाइव को हम नहीं यूज कर सकते. तो DIC microscopy is used for this purpose. Okay, so we have solved total 21 questions. Okay, in the previous video of this series हमने 10 क्वेश्चन सॉल्व किए पार्ट 1 में, एंड इन दिस वीडियो we solved 11 questions. Okay, so if I calculate 21 into 2, जो पार्ट भी 2 मार्क्स का होता है, फुल एक्यूरेसी के साथ it goes to 42 marks. Got it? 42 marks. So this much we have solved. Okay, based on the content available on our channel and previous knowledge को use करके and logic को use करके how we can solve, we have seen. Got it? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to the channel, and stay tuned for the upcoming videos. Thank you.